Okay, welcome back. Now we're going to begin in the following couple of lectures to explore some specifics of protein chemistry, and this will allow us to make some very interesting calculations about how likely it is for the information in DNA and the structure of a given protein to have arisen by chance. And the first concept we want to explore then as we begin this little learning process about proteins is the concept of chirality. And those of you who are chemistry buffs are probably familiar with this, but I would say most of us don't know what is chirality right off the bat. But here is something from a website that says biological homochirality, one of life's greatest mysteries. And that certainly is true. And it is very important to the discussion that we are going to have. We have talked about the Miller-Urey experiment forming amino acids as the primitive building blocks that were in this prebiotic soup of the primitive oceans that supposedly by chance these building blocks began to get together and make functional proteins. So the first thing we need to understand about amino acids is that, like your hands, they actually come in quote-unquote right-handed forms and left-handed forms. The exact same amino acid alanine has a right-handed form known as D-alanine and a left-handed form known as L-alanine. They are quite different actually. They are both alanine, but they are like your right and left hands. So your right and left hands are mirror images of each other. They are both hands, but they cannot be superimposed on each other in the same way. They are reflections of each other. And all amino acids except one come in these two forms, a right-handed form and a left-handed form. And if you've ever tried to wear a left-handed glove on your right hand, you understand that they're not the same thing, right? So biologically speaking, right-handed amino acids are different from left-handed amino acids. And the great mystery of life is that we are homochiral. We rely on only left-handed forms of amino acids. We do not use any D or dextro or right-handed amino acids. All of our proteins use L amino acids. And that is in and of itself a tremendous mystery. Why is that? Well, that is because when a process like the Miller-Urey experiment uses basic chemicals like methane and ammonia and hydrogen, and you run an electrical spark through them, you produce a 50-50 mixture of D amino acids and L amino acids. That is known as a racemic mixture. It has half D and half L. So if these D and L amino acids are floating in the primitive prebiotic ocean soup, as this article says, we would expect that the building blocks of our bodies would have incorporated both D amino acids and L amino acids. Very mysteriously, it turns out that all life forms only use L amino acids. They do not use D amino acids. And sugars can also come in right-handed forms and left-handed forms. And it turns out that we only use D sugars. The right-hand forms and left-hand forms are known as isomers. They are both, for example, alanine, but you have the D isomer and the L isomer. The fact that biology uses only one isomer of amino acids, the L amino acids, and uses only one isomer of sugars, the D sugars, is known as homochirality. And it is an entirely unexplained mystery as to how this could have happened if life arose from the primitive prebiotic oceans by chance combinations of amino acids, since we know that experiments like the Miller-Urey experiment produce a racemic mixture, 50% D amino acids, 50% L amino acids. And when we come to quantitate the likelihood of one of our proteins forming by chance, this idea that we use only L amino acids will become significant. However, even without the math, it is probably a fact that most of our listeners are not aware of. It is a true enigma, a true miracle of biology, happening in our bodies all the time, and not just our bodies, as this article says, from bacteria to elephants, that is unexplained. And of course, in my mind, the best and only way to explain it is intelligent design. But it's worthwhile to know about this miracle nonetheless, the miracle or the enigma or the mystery of homochirality. Now, a second important fact that we need in our arsenal has to do with how amino acids link to make a protein. We've already talked about how a protein is just a long chain of amino acids, sort of like a pearl necklace, and the key issue is that the amino acids have to be in the right order. Now, how do these amino acids link together? Well, if you look at the top right hand of the slide, you see the structure of an amino acid. On the left-hand side, it has a nitrogen and two hydrogens. That's known as an amino group. And on the right-hand side, it has a carbon, and that's double bonded with one oxygen and then bonded to an oxygen and the hydrogen with a single bond, and that's known as a carboxyl group. And the way that amino acids link together is that the carboxyl group of one amino acid will join with the amino group of the other amino acid. So what happens? We remove the oxygen and the hydrogen of the carboxyl group and remove one of the hydrogens linked to the nitrogen on the amino group, and those two hydrogens and one oxygen make water, and the carbon of the carboxyl group will link to the nitrogen of the amino group. This is known as a peptide bond. And all of the amino acids in all of the proteins of our bodies link together through peptide bonds. Now, why is this important? This is important because if I put a bunch of separate amino acids, like the products of the Miller-Urey experiment, into a water bath, they don't automatically form peptide bonds. Some of them will link to each other by peptide bonds, but they can also bond to each other in different ways. And the likelihood that two amino acids will form a peptide bond rather than some other kind of bond is about 50%. So if I want to link two amino acids together and I put them in a water bath, there's roughly a 50% chance that they would form a peptide bond, which is what proteins in our bodies need, and roughly a 50% chance that they will form some other kind of bond, which would not work for our proteins. And this fact will also become significant when we look at the possibility that functional proteins like bacteria use, like we use, arose by chance from the primitive prebiotic environment by so-called chance prebiotic evolution. Now, as we begin answering our question about the likelihood of chance producing a 150 amino acid protein, which once again is one of thousands of proteins which our body uses and is a protein of relatively modest length. I want to give credit where credit is due. The analysis I will be presenting to you is found in an outstanding book by Stephen Meyer called Signature in the Cell. It's in chapter 9, and if you're interested, also chapter 10 of that book. And Stephen Meyer got his PhD at Cambridge University in origin of life research, and he has become one of the leading proponents across the nation and really across the world in the intelligent design movement. And I find his thinking very lucid, very clear. And at the end of this lecture series, inshallah, I will present a brief bibliography of the books that I relied on. And I've already presented you with a lot of articles, of course, along the way. So I've tried to kind of source everything I say, but I would like to give special recognition at the end to a few books, and this will certainly be, very high on that list. Okay, so to give the scope of the amount of information contained in one small gene to form a 150 amino acid protein, let's look at how likely this could have happened by chance in the primitive prebiotic ocean, which has a racemic mixture of amino acids, supposedly, as per the Miller-Urey experiment and subsequent chance hypotheses of prebiotic evolution. So the first thing is that somehow we need to select one L amino acid, then link it to another L amino acid, then link it to a third L amino acid. And if I've selected one L amino acid, the chance that I would do that is 1/2. Then that the next amino acid that comes to link to it is also 1/2. Then that the next amino acid that comes to link to those is also an L amino acid. Well, that probability is 1/2. Each time it's as if I'm flipping a coin. So if I want a string of 150 L amino acids, that is really mathematically the exact same problem as flipping a coin and getting a series of 150 heads. And that probability is 1/2 times 1/2 times 1/2, 150 times, or 1/2 raised to the 150. In more familiar terms, that is 10 raised to the 45, roughly speaking, approximately. So that is roughly one chance in a billion, 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 billion. Now, these amino acids don't just need to be L amino acids, but they need to each link with a peptide bond. So if I have one amino acid and I bring in a second amino acid, what is the chance that the bond that links them is a peptide bond? Well, we said that that chance is 1/2. Then linking to the next amino acid with a peptide bond, not some other kind of bond, well, that chance is 1/2. And the probability of that happening is again 1/2 times 1/2 times 1/2, 1/2 to the 150. Okay, again, like flipping a coin and getting 150 heads. Now, I know some astute listeners are going to pick up the error in what I just said, that actually if I have 150 amino acids, I have only 149 peptide bonds, so it would really be 1/2 to the 149, but close enough just to make the figures round. So again, roughly 10 to the 45. Now, what is the possibility that I have 150 amino acids, all of them L amino acids, and all of them linked by peptide bonds? Well, it's 10 to the 45 times 10 to the 45, and that is 10 to the 90. So the probability of by chance getting a single 150 amino acid protein with all L amino acids and all peptide bonds is 1 in 10 to the 90, or 1 in a billion, 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 billion. So to put this in perspective, if I take a look at all of the subatomic particles in the entire universe, all of the neutrons, protons, electrons, how many of those are there? They are about 10 to the 80, physicists estimate. So the likelihood of forming one single such protein is about 1 billion times less likely than finding one specific subatomic particle, finding the correct subatomic particle by a blind search in all of the particles in the known universe. So that in and of itself should right away put the nail in the coffin of the chance hypothesis, but things don't stop there.